Like I was saying, yesterday we had talked about cell theory. We talked about the scientists involved in cell theory and the three tenets of cell theory, which tell us that all living things are made of cells, that the cell is the most basic unit of structure and function for all living things, and that all cells come from other cells. So that's our cell theory. From there, we had then started to discuss levels of organization. We had talked about how all living things are organized. We talked about how it starts at the atom, and then there's molecules, and there's organelles. But for living things, that level of organization begins with the cell. Enough cells get together, they make tissues. Tissues get together, they make organs. Similar organs get together, they make organ systems, and then, of course, organ systems get together to make an organism. We then moved on and talked about prokaryotes and eukaryotes, and we said that eukaryotes and prokaryotes, the big difference was that eukaryotes have a nucleus and prokaryotes do not. And that's kind of where we're picking up here. Prokaryotes are usually going to be unicellular organisms. And I shouldn't even say usually, they are pretty much 99.999% of the time are going to be unicellular organisms. Prokaryotic cells do not separate their genetic material with a nucleus. There is no nucleus in a prokaryote. The nucleus is what we call not the nucleus. The DNA is just sort of like floating around in the cytoplasm. Now, we do have a cell wall, like in plant cells. We do have a cell membrane, like in all cells. What we do not have are membrane-bound organelles. There are very, very few organelles in a prokaryote. Prokaryotic cells tend to be very, very small compared to eukaryotic cells, and they tend to be a lot simpler than eukaryotic cells because, again, we don't have all the specialization in the organelles happening. As a general rule, we are going to see the eubacteria and the archaea bacteria in these in this category of cells. Most of our conversation about cells, though, is going to be around eukaryotic cells because A, they're more interesting, and B, as a general rule, we have more experience with them because we ourselves are eukaryotic. Okay, so this is a typical prokaryotic cell. We always see a cell wall, a cell membrane, and then we call it the nucleoid region. That is pretty much the stuff that would normally be inside a nucleus, but it kind of like is just floating around. And then it also has ribosomes. And it has ribosomes because ribosomes specifically are needed to build proteins. And we know that proteins are necessary for life because we already talked about them. We talked about macromolecules. Now, not all prokaryotic cells have flagellum, but a flagellum is kind of like a tail that helps move it around. All right, so you now have your Venn diagram showing us the differences and similarities between prokaryotic cells and eukaryotic cells. So we can see for prokaryotic cells, pro rhymes with no nucleus, no membrane-bound organelles. Eukaryotic, U rhymes with do. They do have a nucleus. They do have membrane-bound organelles. All right, so eukaryotic cells can be multicellular, unicellular. Animals, plants, fungus, protists, they tend to be very complex. They tend to be larger. As far as prokaryotic cells go, these are unicellular organisms, meaning one cell only. Bacteria, archaea, they're very, very, very small, and they're not very, very complex. So what are these? But if they're still cells, they have to have something in common. Well, they have in common what all cells have in common. Every single cell on planet Earth, whether it is prokaryotic or eukaryotic, must have DNA, a cell membrane, Remember, DNA is that genetic material, one of those characteristics of life. Cell membrane helps maintain homeostasis, one of those other characteristics of life. Ribosomes are really important in the production of proteins, and we know that all living things need proteins. And prokaryotic cells and eukaryotic cells both have cytoplasm. And we'll talk about what cytoplasm is in a moment. But again, this lovely little Venn diagram will come up again later on in life. So make sure that you have it completed and you have it for reference material. All right, so hopefully you had a chance to pay attention and see some of the differences between prokaryotic and eukaryotic cells. I got a couple things in the chat. All right, so when we talk about cells, ladies and gentlemen, we are going to, again, mostly talk about eukaryotic cells because they actually have organelles. Um, organelles, and we are moving on in the note catcher. Cell organelles, it should be located right underneath the Venn diagram. Cell organelles are part of the cells that have specialized structures and perform specific tasks. Different types of cells are going to have different types and different amounts of organelles. For example, muscle cells, which are obviously involved in motion, require a lot more energy than, for example, a red blood cell, because red blood cells don't even move on their own. They get pumped around by the heart, which is a muscle cell. Um, so muscle cells tend to have way more ribosomes because they have to have a lot more proteins for all that muscle matter. Um, on the other hand, something like, for example, a nerve cell, they may have, I don't know, I'm kind of trying to think about this. What would they have more of? They probably would have slightly more mitochondria, if I'm being honest. And I don't know that for sure. I'm just making a guess. And again, I believe that they would have that because the mitochondria powers the cell, and we know that there's a lot of electrical impulses happening in nerve cells. All right, so we are going to start with the cytoplasm, okay? The cytoplasm, again, eukaryotes and prokaryotes both contain cytoplasm. So what the cytoplasm is literally just this jelly-like fluid that supports and cushions organelles. It's kind of like the gooey insides. Think of it as sort of like the support system inside the cell. Everything in the cell is floating around, if you will, inside the cytoplasm. The cytoplasm, you don't really have anything specific to point at. It's just kind of like the stuff that's in the space in between. Oftentimes when I have students and I'm trying to explain this, I'm like, well, think of like a fish tank, right? A fish tank has the fish and it's got the plants and it's got the little structures, but all of it is connected and all of it is sort of held together, quote, quote, if you want to think of it that way, by the water, right? So the cytoplasm is like the water in a fish tank. It has everything in it. That is what the cytoplasm does. It functions as a cushion. It supports the organelles, and all the organelles and all the stuff inside the cell is in there. Again, all cells have cytoplasm. And a lot of the cellular processes, protein synthesis, glycolysis, a lot of our chemical reactions that take place in the cell happen in the cytoplasm. So cytoplasm, even though it's not, really super exciting, a lot of cool things happen there. Now, next we have the nucleus. I do want to remind you that this is only found in the eukaryotic cells, okay? There is no nucleus inside a prokaryotic cell. Remember, pro is no, u is do. So this is like the control center of the cell. If we were comparing a cell to like a school, I would say that the cell is kind of like the main office, okay? It has all the information. So the cell's nucleus contains the DNA. It has all the information, because remember, DNA, genetic code, that is all the instructions for making you, for making the tree, for making a bunny rabbit. All the proteins and all the other molecules that your body needs to function, to grow, to be, all of these instructions are in the DNA. And the DNA is found in the nucleus. The nucleus is is surrounded by a double membrane, which makes sense. And DNA needs to be protected. And you have these small little openings in the double membrane called the nuclear pores, which those come into play later when we talk about how DNA and RNA and proteins are created. So we'll get to that eventually. All right, so we have vacuoles and vesicles. Okay, vacuoles and vesicles. Vacuoles are like they're like storage containers. They literally just store things like water, salt, proteins, carbohydrates. Um, Plants tend to have very like a single large vesicle. Animal cells have many small vacuoles. Um, in plants, a single large vacuole will actually serve also as a support system. I realize that you guys are probably too young. Well, I don't know. Maybe you do, maybe you don't. If you have any memory of this whatsoever, it was a much bigger thing when I was younger. There used to be this thing like people would love having like their water beds. Don't ask. It was a thing. It was the 70s. It was a weird time. Anyways, um, if you didn't have enough water in the water bed, like you would, get on the bed and you would like feel the structure underneath, which sucked. So these large vacuoles kind of work like water beds. So when there's enough fluid in them, it helps to support the structure of the plant as well. Much like if you had enough water in your water bed, it would support the weight of the individual who, got onto the water bed. A vesicle, on the other hand, is like they just move stuff around. They can store material, but also they move things around between organelles and also through the cell's surface. So if things need to leave the cell, they leave in a vesicle. If they need to come into a cell, they come into a vesicle. And if I have something that has to go, for example, from like the chloroplast over to the mitochondria, it's going to travel there in a vesicle. All right, lysosomes. Lysosomes, again, we only find these in eukaryotic cells. They are a membrane bound organelle. Lysosomes are little tiny, itty bitty small organelles, and they are filled with digestive enzymes. They break things down. They break down lipids, they break down carbohydrates, they break down proteins into small, itty bitty molecules that then those molecules can be used. We talked about this previously when we talked about dehydration synthesis and hydrolysis. That's what's going on here. We're breaking things down. Also, lysosomes, again, going back to the analogy of the school, will also break down worn out organelles. They are acting like sort of like the custodians of the cell. If something's no longer working, if something is kind of like not functioning properly, they will literally digest it. So they will clean it up and get it out of the cell. They just remove kind of like the junk, okay? Because if there's too much junk that clutters up in a cell, it's going to become toxic to itself, and then that you're going to have, cell apoptosis, which basically means the cell is going to die. All right. Additionally, we have a cytoskeleton. This is found in eukaryotic cells. Cytoskeletons are protein filaments, and they basically give shape and organization to a eukaryotic cell. Used in transport, those little vesicles will travel along the cytoskeleton. It also allows the cells to move and engulf other particles. So, if you recall from our intro video yesterday, one of the things that we saw in the video was this white blood cell that literally looked like it had like shaped itself around and swallowed a virus. The cytoskeleton is what allows this to happen, much like your own skeleton, which like you can you can anchor your muscles to and then helps you move around. Same thing is going to happen here. It's anchored to different parts of the cell, and then when the cell needs to move and do things, it can then, of course, move around. There are two main types of protein filaments in the cytoskeleton: the microfilaments and the microtubules. The microtubules will come very much in handy when we talk about cell reproduction, but that's for another time. Microfilaments, again, thread-like structures made of actin. That's a protein. And it helps to support the cells. It helps the cell move. Very important if you're a single-cell organism, because again, you're going to have to move around on your own. The microtubules are structures made of tubulin proteins. And again, it helps to maintain the cell shape and helps with cell division, which of course is cell reproduction. Now, we have centrioles. These, by the way, are only found in animal cells and they are only found in eukaryotic cells. Centrioles help organize cell division. The reason why we do not see a lot more, I guess, mutations than we do in a lot of organisms is because of these centrioles helping in cell division. They're very, very key to cell reproduction. Ribosomes. Um, all cells have ribosomes. Prokaryotics have ribosomes. Eukaryotics have ribosomes. Ribosomes basically help to build protein. And we already know that proteins are 1,000% essential for life. So no proteins, really, that's one of the four macromolecules. You have to have all four macromolecules for life. If we don't have proteins, we don't really have life. Ribosomes are responsible for building proteins. We can find these, like I said, in all living cells. Oh,
Please excuse this interruption. For the students who have tickets to the OTEC presentation, please report to the media center when the first hour bell rings. Go straight to the media center when the bell rings. Again, for the students who have tickets for the OTEC presentation, once first hour bell rings, report immediately to the media center. Thank you.
I apologize. There was an announcement here, and I had to kind of listen to what they were saying. Okay, so they basically link amino acids together, and so our dehydration synthesis, when we are building proteins, when we are putting these things together, this takes place on the ribosome, or in the ribosome, I should say. Endoplasmic reticulum. I am never actually going to spell out the words endoplasmic reticulum 90% of the time. I'm going to call it the ER. The ER is an internal membrane, and ER is responsible for putting together lipids. So remember, again, lipids, these are one of our macromolecules essential for life. We see the lipids being put together inside of the endoplasmic reticulum, specifically the smooth ER, because there's two ERs. There's the smooth ER and then there's the rough ER. The rough ER tends to have the ribosomes attached to it, so we see the protein production there. Smooth ER, lipid production. A little bit more about where are we on time? We have about a minute. Let's finish ER and then we won't move the Golgi apparatus until tomorrow. The rough ER, again, has ribosomes on the surface. Rough ER is where we are going to be making the proteins because that's where the ribosomes are. And you can see how it's sort of set up like channels. There's also movement happening throughout here. Smooth ER, like the name suggests, it's pretty smooth. No ribosomes. And again, the smooth ER is going to be creating lipids as well as detoxification. Your liver, a lot of smooth ER. Our liver in our bodies is like the single largest organ that helps to detoxify the body. So if we look at liver cells under a microscope, so we were talking about specialization earlier, we see huge amounts of smooth ER inside liver cells because again, the entire function of our liver is detoxification. So if you were ever to consume, and hopefully none of you, but most drugs and alcohol, when they go through your system, have to go through your liver to clean themselves out. This is also why oftentimes we see in people who are alcoholics and have drinking problems, or people who are chronic drug users, we see a lot of those patients with huge amounts of damage to their liver because literally their liver is being used so much more than it would be normal for anybody who doesn't have an addiction problem. There are other diseases, of course, that can affect your liver, but most commonly we see damage to the smooth ER, damage to the liver being done by overuse of drugs and alcohol. Anywho, there's my little public service announcement for the day. We are going to stop here at endoplasmic reticulum. We are almost through these notes, which is fine. Tomorrow we will wrap up the notes. It shouldn't take us any more than 5-10 minutes.